Hello, everyone. I'm Kelly Eversole, the Executive Director of the International Alliance for Phytobiomes Research, and I want to welcome you today to our webinar. And we're pleased to have Jeff Bennetzen with us today from the University of Georgia, and he'll be talking to us about host microbe genetics. But before we get to his part of the webinar, I'll want to explain a little bit about the Phytobiomes Alliance. So we are a nonprofit consortium of industry, academic, and governmental scientists, and we are sponsored by a number of different organizations and everything from industry to academia to research institutes as well as governmental research institutes. And I want to thank them now for making it possible for us to have this webinar series as well as our other coordinating activities. So phytobiomes is the complex systems of plant-based agriculture. It's the whole concept of a plant in a particular biome or having a biome and what's that plant actually look like with regards to what's the best plant, what's the best genetics of the plant, as well as all of the interacting aspects from microbiomes, which is one component of a phytobiome, climate, weather, water, but it's really all the biological and geophysical components that affect a particular biome and how we can have the most sustainable plant in that particular biome. And of course, it's all influenced by management practices as well. So the phytobiome is really looking at that entire complex system. Our vision is that by 2050, all farmers will have the ability to use predictive and prescriptive analytics based on geophysical and biological conditions for determining what's the best combination of crops, the best management practices, and the inputs for a specific field in any given year. So our next webinar will be in about a month, and will be focused on sustainable agriculture, kind of quality management, a follow-on to some of the things that we're talking about today with regards to our Arbuscular mycorrhizae fungi, but in this case it'll be regarding inoculants containing AMF. So you can just please note that the time is different because we have our presenter is from Australia, so we will be doing it at a different time. But as always, it'll be on our YouTube channel. I also want to mention that we will be having, again, our Phytobiomes, Exploring Phytobiomes workshop at the Plant and Animal Genome Conference. And abstracts are now being accepted for that, so anyone can submit an abstract and be included in our workshop. The deadline for submissions is the 12th of October, and we hope to see you at that one. Just to remind you, the webinar is being recorded and will be posted on the Phytobiomes Alliance YouTube channel shortly. And you can subscribe to the channel so you never miss an upload, and there's a link for you to do that. The presentation itself will be followed by a Q&A period. Please submit your questions in the Q&A panel, but also monitor the chat panel because you'll see messages from the organizers, as well as if Jeff has mentioned a paper, there will be a link to that paper on the, often there will be a link in the chat. Already you can download both a PDF of my presentation, which will have the links, as well as Jeff Bennettson's presentation. It's in the handout section. So without further ado, I would like to introduce Jeff Bennettson. I've had the pleasure of knowing him for 28 years. I counted how long we've known each other, and going back to establishing the maize genome sequence many, many years ago. and it's been a great pleasure to work with him over the years. He's at the Department of Genetics at the University of Georgia, and he has done a lot of work in transposable elements and focused a lot on plant genome structure and evolution, but particularly as it relates to transposable elements. And he is a member of the U.S. National Academy of Sciences and also a fellow of the American Association for the Advancement of Sciences. So we want to really welcome him for joining us today to talk about host genetics and systems biology of arbuscular mycorrhizal fungi and other sorghum root microbes. So Jeff, thank you for being with us today, and I'll turn it over to you.
Thank you, Kelly. It's certainly a great pleasure to have a chance to talk with this group. As you pointed out, this is not a field I've been in for as long as some others, although I've been working in the science biz for about 40 years, a bit over 40 years now. And actually about a decade ago, I started moving into microbiomics at a certain rate. Most of my work before that was on plant genome structure, function, evolution, and particularly as driven by transposable elements. And to tell you the truth, I found that about a decade ago, most of the things I was interested in, we understood pretty darn well. And so I was looking for something that had a lot of questions that needed to be answered, and certainly microbiomics came to light as an area of research that had a whole bunch of unanswered questions and with technologies that would make them very answerable. I didn't have any funding to work on this. In fact, I first started working on it about 2002, so it's like 2 decades in this, but just a little bit of myself in my spare time when I wasn't teaching or doing research on genome analyses. And we started publishing a few years later, but it's really been dribbling out a bit here, a bit there over the last, I guess we've been published for about 15 years now. So the last little bit of publishing of papers here and there. And because I didn't really have any funding to work on this, I didn't want to ask for funding because I was still very well funded to do my genome stuff, and I didn't feel like double dipping that way. The other thing about this was I didn't really feel comfortable, because I was interested in the host genetics, with the timeline of giving these kind of projects to graduate students, because to do a genetic analysis of this type would take, just for the genetic parts on organisms like maize and sorghum that I work on, 5 years or so, which made it really sort of dicey time-wise for a graduate student to get involved. So we basically decided about 4 or 5 years to get very seriously into it, and I wrote my first proposal, and I'm going to talk about that. It was funded. In fact, I'm now 1 for 1 on proposals in the area of microbiomes. I wish my frequency or success rate was so equally high in any other field I've worked in. But and I'm going to talk a little bit about it. We're just in sort of the beginning of it, but it shows, I think, the thinking behind the kind of science we're going to do, and it has a lot to do with the kind of thing that Kelly described in the phytobiome analysis. So we're interested in host genetics. So I'm a plant geneticist. I'm not a microbiologist. For me, microbes are massively interesting, but most of the time they're actually a phenotype that I'm studying. They're the phenotype to, in fact, map and identify plant genes, and the plant genes that determine which microbes are present in a plant microbe interaction and what those microbes are doing. Now, the microbes, of course, determine this themselves, but they do this in an interaction with the plant, and the plant has a major say in this. Our first sort of significant publication on this a few years back was showing that farmers had farmers in working with foxtail millet had actually selected for microbial populations in the roots without knowing so, but they had different microbes that microbial communities associated with domesticated plants compared to the wild plants, and that was a selection, an inadvertent outcome of their selection for higher productivity and other domestication traits. So we knew that genetics was going to be a big factor, along with lots of other factors that you all know about, like soil type and other aspects of the environment. So About a decade, about 2 or 3 years ago, we got funded to do a big project working on arbuscular mycorrhizal fungi and its association with sorghum, and I'll talk significant about that in a second when I first tell you the sort of the foundation for why we decided to propose that. And our overall intention is very appropriate from the phytobiomes perspective. We want to develop a systems biology data set framework for understanding how all these factors that impact the plant microbe interaction are involved and how they affect crop productivity. And particularly, as I say, we're interested in identifying the most beneficial for any given environment, the most beneficial plant genes. Now, let's give a little bit of information that those of you that don't know a lot about arbuscular mycorrhizal fungi. There's 21 genera in this phylum. It's an ancient phylum. The number of species that have been proposed so far is about 3,700. We think that number is probably several fold, maybe as much as a order of magnitude too low, because every time we look at a sample, we find things that would be called new species, make up over half the material we're looking at. It's an ancient association. You see it in the roots from some of the most ancient land plants, and pretty much almost all land plants have that now. Not all. There are some, the Brassicaceae, for instance, do not have this, and it looks like from analysis one can do phylogenetically, it looks like it was a trait that was lost, and there's reasons why you might want to lose AMF associations that we'll talk about in a second. It provides host plants with a number of useful values. The most famous is phosphate uptake. It also is very important for water uptake in drought conditions. A lot to do with nitrogen balance. In some instances, it's been shown to affect disease resistance for both as a biotic stress as well, and decreasing access of pathogens to root tissues. So this is sort of a simple diagram of from others, Thurkel et al., and showing on the left here a root with these little fuzzy blue things being circular blue knots here being mycorrhizal materials inside the root. A lot of the mycorrhizal biomass, probably the great majority of it, is outside the plant, out there collecting. And in fact, there's a lot more surface contact with the mycorrhizae than there is soil contact, than there is with roots or root hair. So it's actually just massively extends the root. It connects plants to plants. There's information transferred, sometimes nutrient transferred from one plant to another. There are ghost plants, for instance, that use orchids that use AMF as a way to get food from their host. They don't themselves attach to the host, but they they attach to an AMF, which is attached to another plant species that provides them with the carbon that the AMF gets, and then the ghost plant can grow by that strategy. Now, the root system has the ability to take up phosphate, but it doesn't extend very far, and it isn't particularly good at it. AMF's much better, and so you see this phosphate depletion zone where the AMF is digging that out, and there's also lots of other growth that's going on. You can see AMF in the root. So here's some root uninfected and infected with AMF, and vesicles, arbuscules, hyphae, intraradical hyphae. So you get various structures you can identify, and as I'll talk about very briefly, we've developed automated phenotyping platform that allows us to characterize the overall abundance and the various types of infection of a given root sample. So the most important thing for our project that I think we need to take into account is the contributions of AMF, like those probably of every other microbe, but certainly it's true for AMF, are context dependent, and they vary dramatically. There are many cases where AMF can actually be parasitic. If you're growing plants on high energy soils, that is, that have got lots of nitrogen, lots of phosphate, sufficient water, then AMF, which the plant is feeding with various fixed carbons, including fatty acids, the plant is feeding these, and this actually just decreases the rate of plant growth to have AMF present. But in other circumstances where any of these characteristics in the soil or in the environment that AMF helps uptake, then it's wholly beneficial. So this range of beneficial to parasitic not only varies across environments, it actually varies to its degree across species and in association with other aspects of the biotic and abiotic aspects of the genome of the microbiome and the genetic environment that we're in. So we hope to dissect a lot of these connections and their conditionality in the project I'm going to talk to you about. Now, we decided to look at this project, to propose this project was based on some work that my colleagues, Katrine DeVos's lab and mine had done on a biofuel crop called switchgrass. And this is just some of the results you get. You see a lot of publications on AMF talking about AMF does this and AMF does that, and they look at AMF in general. We find in our experiences, it's rare that two AMF of different types do the same thing, even when they're present in the exact same location, and that's probably why you have more than one present there. So we think if you're describing AMF behavior in this sort of mixed environment, that you're probably describing something that's not particularly useful in many ways. So this is a single root that we took of switchgrass and then did analysis of amplicons across an informative molecule for AMF. And these are the different ecotypes we found, or taxonomical units that we found across here, which could be called species, although that might be taking it a bit far. And as you see, we had a lot of Rhizophagus in this particular environment here in Georgia, quite a few Glomeromycota, quite a few Phenoliformis, some Septiglomus, some Paraglomus, and some from a genus that hasn't yet been given a name. And so we got quite a few different types here. And as I'll tell you in a second, we get different behaviors from each of these different types. So if you do an analysis that only looks at AMF per se, you're averaging those out. So if this one's up for a second property and this one's down for a second property, the overall look may look like it's down because you've got so many of these, but this may look like, and therefore the contribution that this makes may be missed completely. We also find in cases, I won't talk about those today, that in fact closely related members of the same genus can behave quite differently because they aren't the same line. They're in fact different, possibly species, if you want to use that term. So a lot of diversity out there. And so all of our analyses look at the genus level. We never talk about AMF. I'll give you an exception to that later on, but we never talk about AMF. We always talk about a genus of AMF, and we'd like to talk about species of AMF, but the complexity and the challenge, the cost, as it were, of actually determining individual species is one that we can't really absorb at this time. So a lot of diversity, that's the first thing we knew. Other studies had shown they're host species specific. We also showed, as others have shown now, but we showed this 10 years ago when we did this work, that there's a genotypic specificity within the species. So this is two different families of switchgrass. Switchgrass is an obligate outcrosser. It's a polyploid and it's a perennial. So you don't have any inbreds for switchgrass. So any particular switchgrass plant, when you cross it to another plant, both parents start off massively heterozygous at various loci. And so you get families that are related, but they're not exactly inbred. So this is one family, 317, which we call our high AMF family. And this is another family, 458, which we call our low AMF family. And these are three plants. And what you see is these 111, that's four different tissues from the same root. 222 has three different tissues collected. This has three different tissues. And then they've been analyzed for the amount of these various different AMF. And as you can see, this particular family has quite a bit of Rhizophagus, but not universally. This one has a lot more of a particular unclassified genus of Glomeromycota. And so, so this was our high AMF population, if you look at overall AMF, but it's actually quite low for some, like Phenoliformis, for instance. Whereas this one, our low one, has a lot more Phenoliformis, and it has a lot more Paraglomus, but it has a lot less Rhizophagus. So if you add it up, once again, you get less here overall, but for different categories, this is high for a few and low for others. So really, to say high and low is somewhat misleading, especially because our eventual analysis is going to be done a genus by genus anyway, so we don't have to use these high and low terms. You may notice that the differences between the different root types, fine roots versus mature roots versus deep roots versus shallow roots, is pretty low. For individual plants, we get, at least at a later point in development, we get pretty much the same values. And that was a shock to us that different root tissues gave us pretty similar amounts of AMF. But that's in fact what we've seen. I'll give you an example of that later on with sorghum. So when we did this association, we were able to use the amount of AMF present at a genus level to perform, I've already talked about these first two, this third one here, to map QTL. So we mapped QTL for AMF abundance, but by using the genus as a level. And in doing that, we found 7, this is on a couple hundred individuals. We call these individuals pseudo F1 and or pseudo F2 because you cross one individual to another, two parentals, because they start off heterozygous, all their progeny then segregate for a trait later on, or in that first cross, which will then segregate subsequently as well in subsequent crosses. But in the first cross, it segregates. So even though it's an F1, it's because it's segregating, you can actually map it as if it's an F2, and you end up with a map for the male and a map for the female because you've got different traits segregating there, or different polymorphisms segregating. So anyway, we matched these 7 switchgrass QTL, and they're all, none of them overlapped. So there was no case where 2 of them were the same place. And we got this for 5 genera of AMF, and the contributions were not small, 8% to 23%. But of course, this is not a huge population. It was less than a couple hundred. And so the smaller your population is, the fewer QTL you find, and the higher their apparent impact seems to be. So if we had a bigger population, I'd expect we get many more QTL, and they'd be a somewhat smaller individual impact. Now, what this tells us is that there are particular genes in switchgrass, in this case, that attract or support or block the removal of some combination that leads to the accumulation of particular genera of AMF, and that these genes do not act on other genera of AMF. They're genus specific. And to emphasize this is when we took the AMF total data and used it also as a phenotype to do mapping, we got no QTL at all. So having the individual genera having different properties with working with different genes basically meant that that created noise to make you miss any QTL if you looked at total AMF levels. So that was a justification. If this happens in switchgrass, maybe this happens somewhere else. And we knew it was true for rhizobium. I won't go into the details of this, but we looked at rhizobium and switchgrass as well. And this one I like to show because we had the same switchgrass that was grown in Athens, Georgia. Because it's a perennial and you can't get identical lines by genetics, you basically cut out, you divide the plant and transfer them and plant them in a second place. This had been planted 5 years earlier in Ardmore, Oklahoma, and it still had the identical rhizobium. This is the endophytic values. Identical rhizobium in both locations. So if you draw this, you get a straight line between them. So that's one thing. These are the 2 parents with that cross. And it's interesting, not only do you get a nice identical, R squared is like 0.9 here. This is a really good correlation between the 2, but you also get massive transgressive segregation. So if you compare the rates in Georgia, you get about 7-fold higher for this individual here, amounts of rhizobium in the root, whereas if you compare this at Oklahoma location, it's about more like 50-fold higher. So you get much higher levels, lots of transgressive segregation for when you plot the individual progeny and the amount of rhizobium they have at these 2 locations. And you can do this once again to map QTL. This was one particular QTL for this, not particularly massively strong, but met our thresholds for this particular experiment. So we decided to do a similar project, but to do it in a lot more detail and to do it on sorghum. And the reason we use sorghum is it's just a vastly more important crop for lignocellulosic biomass production because it's more improvable. It's not only a major biomass crop, but all over the world, it's an important crop for dryland agriculture, and particularly vital in the developing world, places like the Sahel and more drought-prone regions of Southeast Asia, etc. So it has high yield under low inputs on poor soils. For our purposes, the fact it's a diploid makes it really useful. We've got inbreds and we could do crosses easily. It's highly diverse. It's got excellent genetic tool set, including lots of sequenced genomes, RNA sequence data, mapping populations that have already been mapped, diversity panels, and particularly for us, it's got sequence indexed mutagenized stocks, so we can find sorghum lines. If we have a gene we're interested in, we can go to a database, find a sorghum line that's got a mutation in that gene, and then do testing of the biology of that mutant line. It's got an active research community all over the world. It exists in both perennial and annual forms, and I won't talk about it, but we're actually looking at perennial versions of this. There are segregants out of some of our samples that are annual, so we're looking at fairly isogenic comparisons between perennial and annual behaviors. And also there are sweet stalk sorghums that are extensively used in some parts of the world right now as a biofuel crop, because you can use it the same way you can use sugarcane. And in fact, in Brazil, it's very important now because it matures at a different time than the sugarcane does. And so these sugar refineries that they have there lie empty a lot of the year, at least they used to, but now they can take the material from sweet stalk sorghum, and that extends the yearly use of those facilities and therefore their profitability. So as I mentioned, pretty highly variable. I won't mention show you the field pictures, but these are just head types, seed color. And the material we got for our analysis is 385 members of a GWAS panel that Steve Kresovich had developed. And these, this is just a structural analysis of the, you can see there's basically 6 races with a certain amount of crossing in gene flow between them. And those have already been mapped and characterized for a number of traits, but not their soil microbiome. So our goals are pretty simple for this. We really want to find out the genetic basis by which sorghum genotypes cultivate specific interactions with AMF. And we want to know the host genetic basis, but we also want to get the AMF genetic basis at the same time. It's going to be an outcome of our research, we expect. And we're actually looking at the entire rhizosphere community because there's no way that doesn't affect the AMF in dramatic ways. And we want to know how this process occurs over different times and environment. And we'll talk a little more about that. Now, there's a lot more goals, and I just put these in here as slides because if you want to see what this big DOE-funded project looks like, but I'm not going to go over them. I basically just summarize them there. And I will talk about the hypotheses we're behind, though. And one of them is that there are sorghum genes whose allelic variation determines AMF abundance and performance, and that we can map these genes. And that's just what I showed you we did in switchgrass. So we assume it's going to be true in sorghum as well. We also assume one of our hypotheses is that the microbial environment influences which sorghum alleles generate the most beneficial AMF symbiosis. And of course, that microbial environment is other microbes. It also includes the AMF as well. Our third hypothesis is that abiotic variables are going to help determine which sorghum genotype interactions are optimal. Right. And so they're not going to be the same associations in each case, although if they were, that would be cool, but they aren't likely. And then finally, in systems biology, we're going to do a analysis of all this at a systems level, so that we believe that we'll be able to generate models that are predictive. And the purpose of these models is to put these data together. Our original work is all going to be descriptive. Most of what I'm going to describe to you is purely descriptive stuff. But in the last couple of years, we're going to be using the correlations we identified and testing them genetically to see if those hypotheses we generate from those correlations turn out to be true. And so here's simple systems analysis. We're doing phosphate and nitrogen analysis, and really we can only look at its effect upon the amount of taxa that are present. It's not just AMF of the entire microbiome present. We'd like to do that with our RNA-seq and our eQTL analyses, but we just can't afford to do RNA-seq on all the thousands and thousands of samples here. We are doing image analysis on the thousands and thousands of samples. We're doing plant phosphate nitrogen analysis, for instance, on retained. We're looking at using this in the genotype we know to look at the QTL that are generated. So, and as I mentioned, this creates a model that is going to be based on correlations. But then as we decide, okay, here's a particular gene that we map that seems to influence which AMF is present here and what that AMF does. Our prediction now is if we have a mutation in that gene, and now we're just comparing mutant to wild type for that isoline. If we have a mutation in that gene, that sorghum, that AMF line with the mutation, the AMF will not be associated as abundantly with the line with the mutation, or won't perform the same functions. So we can test these in a hypothesis-driven way once we've got these correlations. But the number of data points to fill in this systems analysis could be in the hundreds of millions, but we're starting off by a rather small sort of hundreds of thousands data points. But of course, that means each microbiome for us is one data point. So if you count that, the complexity in each our microbiome sample analyses, then really we're generating trillions and trillions of data points. So as I mentioned, years 1 and 3 are just descriptive and model building. Years 4 and 5 will actually take these. And then I've got some examples here of the kind of experiments we might do to see how this works, see whether our correlations are just by chance or actually have a functional basis, causal basis. So the major variables we're looking at are genotype of the sorghum, those several hundred GWAS lines for the GWAS analysis. Field location, we're doing the work in both Georgia and Arizona, we're doing identical experiments in both locations. Fertilizer input, nitrogen and phosphate, and we're doing one drought experiment in year 4 in Arizona only. So once again, there's proposed experiments, and I'm not going to go through these. I just put this slide in once again so you'd see what we're actually going to do if you want to look that up, or you could look at the grant proposal at DOE, and that's available. So just another slide of that. And we have an ultimate goal to this, and it's, I think, in agreement with what the Phytobiomes Alliance is after, but it's very different from what you're going to hear in the talk next week. So our goal is to create the correct, to identify the right sorghum genotypes that will attract the appropriate AMF and support it, that they will not, no one will need an AMF inoculum because it'll find the AMF it needs to give the most beneficial outcome, and that most beneficial outcome will be a decrease in needs for such inputs as irrigation and fertilizer. So, so our approach really is not to find new and better AMF, it's to find the plant genes that attract the best AMF. Okay, so the first thing in the 2021 and 2022, when we first started these experiments, we've been doing time courses, and mostly during the period where there's a mature root system. So I'll talk to you about that. We didn't look at, I've got another project with an undergraduate, a graduate student working with an undergraduate in the lab to look at a time course on the order of hours to days, but after the root gets in the soil. We're looking at the full microbiome. We're not just looking at the AMF, although we're concentrating on the AMF, and the data I'll give you today is really just about the AMF in that situation. But we're looking at the entire community by in some cases, we're doing amplicon sequencing. In other cases, we're doing shotgun sequencing. Shotgun sequencing is just too expensive for us to do it all that way because of library costs, construction costs. We hope that we'll do as much as we can with shotgun eventually. And we're going to describe both the plant and microbiome genes that are expressed during this interaction under these different circumstances. And as I mentioned, we've automated the microscopic scoring of the amount and status of AMF and roots. The ratio of arbuscules, for instance, to vesicles is a measure of, considered to be a measure of the productivity of an AMF interaction with a root. So here's our root structure sort of experiment where we've got 8 compartments, 4 depth and 2 across, distance from the center of the plant. So this is our root center, root periphery, and then these 4 different depths. And also because there are things like, for instance, fine roots at all these depths, we made special samples of fine roots as well. And we said, okay, if we take at a given time point, the microbiomes from each of these, how different are they? And the first analysis was just for AMF, and that's what you'll see here. And what you see is the AMF communities for all these things are the same. We don't get any difference at all at about 70 days when this was sampled post-planting. So, which is good for us. It means we can sample these, and we're most interested in AMF. We're analyzing this data to see if the rest of the communities change. I'd be surprised they didn't, but at least the AMF isn't. So we know we can sample the AMF at these times, given times, and we don't have to chop off little bits of root here and there. We can take the whole root system, and it's going to give us pretty much the same result. The communities I'll tell you in a second were really quite different from Arizona and Georgia. That isn't shown here, but at those locations, once again, the root type didn't seem to make any difference. Now, this is just a table here showing what we did find to have significant impact. Turns out the time point at which we analyzed, but they're all between 50 and 90 days post-planting. They weren't early on. Data I'm not going to show you shows early on the AMF communities changed dramatically, but after about 50 days they seem to be pretty stabilized, and there is some difference in those communities, but it's not dramatically different. It's not statistically significantly different, and this is good for us once again, in that we can harvest plants at different times and say we're getting pretty comparable results. Of course, one of the factors we enter into this huge systems biology is number of days post-planting that we do analyze it, and we also have to analyze the number of days pre-flowering, because these flower at different times. Some of them don't even flower, and these are the ones that the non-flowers, of course, create the greatest biomass. So we have to write down the date and the developmental status of the plant and various other factors that we score and add into our systems biology diagram. But the two that did seem to make a huge difference was the genotype, and even more important was the site. The site was huge. So this is satisfying. The genotype was significantly different. We only used in this is our first mini experiment, only used about 10 different genotypes, not 300 some. And so this was feasible to do this easily in a fairly short period of time. And so the genotype had a big difference, but even more important was the location. Where we grew it, Arizona versus Georgia, was hugely different in the populations. And I'll just show you a little bit of analysis of that. So this is a sort of a summary of the results I'm going to show you down below. This is a linear discriminant analysis, which is a kind of a principal component analysis in some ways. It's basically a way for continuous data for looking at emphasizing the dramatic differences. And so in Georgia, we got a lot more of these six genera, sorry, of AMF, whereas in Arizona, we got a lot less diversity, and we also got a lot less evenness, and we also got a lot more of diversa spora. And this data down below, and you'll see some like Scutellospora was present at both locations at fairly similar levels, whereas the Archaeospora at Georgia was 70-fold in a relative sense, 70-fold more abundant than it was in the Arizona. So big differences in what ones are there, but as I mentioned, they all seem to reach maturity as far as instability of that community within the first 50 days after planting. So now we have the first year we just did this summer of the big field, and the big field was big. So this is 12 samples here, and each of them contains all 350 plus genotypes, 6 plants of it, and we'd harvest 2 plants from each, so little groups of 6, and we harvest the middle 2. And they're all, there's just 3 replicates of different treatments. So these 4 are 1 replicate, and there'll be one that is high phosphate and high nitrogen. It's been fertilized. There'll be one that wasn't fertilized at all. There'll be one that received only phosphate and one that received only nitrogen. And then that same pattern is repeated at these other 2 locations. So we have triplication of these samples, 4 treatments, high N, high P, high N plus P, and then no added fertilizer at all. This is Georgia, so we don't worry too much about water. We did have the capacity to irrigate if we needed to, which we really didn't need to very much. And so it took us a team. We had a team this summer of about 25 people who did the harvesting mostly undergraduates who did the harvesting, washing, labeling, all this. The labeling here is the key issue that you have to really worry about. So these are massively labeled multiple times and cross-checked multiple times to make sure we've got the right samples. And then the roots from these are going to be scored for AMF abundance microscopically. They're going to be tested for, in some experiments, for RNA expression. That was even a different type of analysis. We had to go out in the field again with another group, because the time frame on that is you have to be really quick before the RNA changes and isolation. And then and of course, we'll look at the microbiome, both the AMF by using amplicon sequencing, but also the entire microbiome by using some amplicon analysis. But also, when we do amplicon analysis, to look at microbiomes, we always look both at fungi and bacteria, and usually look at archaea as well. We don't always look at protists or animals, but we sometimes get them if we're using the fungal amplicons. They often will amplify those up. I'm not sure how quantitatively, but we see them. So we'll get all that information, and that's what will go into this systems diagram that I've talked to you about. And as you can see, the number of data points here is going to be rather enormous, and we'll be able to see what correlations show up. And really, we're interested in any correlation, because any correlation, the tighter it is, it will rise up to the a level of something we want to predict, we want to use as a hypothesis in the next years 4 and 5 of something we want to test in that hypothesis-driven experiments. So the people, that's what I want to talk to you about today. The people doing the work are basically Philip Brayley Jones, which is right here in the back. Phil is a really experienced postdoctoral fellow who's really good at AMF and population genetics. So he's sort of in charge of most of the microbiome sort of general analysis on this project. Srini Chalavati did all the switchgrass work that I talked to you about later on. That's Srini over there. Olivia Asher, Josue Fernandez back there next to Phil, Ben Long, and are all people who worked in the field. Pom Pendergast is a senior postdoctoral fellow who also did a lot of the field research. These guys, all these, Olivia and Josue and Ben are all graduate students. They all have projects related to this. Ahmad Kabir is a visiting scientist from Bangladesh, who real expert on fungal plant interactions, although mostly on trichoderma, not AMF. And Tom Pendergast, I said, is a postdoc in Katrine DeVos's lab. This is Katrine DeVos. Most of these other people work on other projects, so they weren't having to do with genome analysis, for instance. Except this is Emma Bennetzen here. She's basically the one person who works on the AMF project that all the undergraduates would basically take seriously, because she was the only one that could talk back to Dr. DeVos and Dr. Bennetzen. So whenever we said to do something, she'd say things like, "Oh, don't take them too seriously." So that was that kept us all grounded. So as I mentioned, a lot of the work I talked on switchgrass is with the DeVos lab. All the AMF work we're planning is with the combination of labs, the DeVos laboratory. Katrine's lab and mine are both plant genetics laboratories, which she's taking lead in the RNA-seq analysis. She's much better at EQTL type work than I am. And she's going to lead the GWAS stuff. The population genetics on the AMF and those kind of correlations are more stuff my lab will lead. Jonathan Arlow's lab is leading the microscopic and he's also analysis and also the systems biology. Annie Chung and Nancy Collins Johnson, are both experts on AMF. There are 2 AMF stars who are doing actual experiments now and will be doing more as our correlations get up. But they also work in the field and in our data generation process. But they'll be mostly doing smaller scale experiments, testing the hypotheses that our analyses will generate. And this Sorghum AMF project, as I mentioned, is all funded by the Department of Energy. The other work was funded on switchgrass, was funded by a Giles professorship. And I got 1 year of funding actually to work on this AMF sorghum, AMF switchgrass analysis from another DOE competitive grant, a big one that's part of the Bioenergy Resource Institutes. I got 1 year of funding to do that, which is what Srini did his work on switchgrass. So that's all I have to say for now. And thank you for listening, and I'm open to any questions you want to ask.
Thank you very much, Jeff. So it's a really interesting project that you've described, and an extremely complex project, no less, but it brings up a number of different issues that are certainly relevant to our efforts in the overall Phytobiomes Alliance, and how we're trying to begin to look at developing systems that can provide us and provide growers with the predictive tools that they need. So just a reminder to the audience, you can put any question in the Q&A panel at this point, and we'll try to get to them as we move forward. So one of the things that you talked about was the contextual aspects of AMF and the contextual aspects of the host genetics. So with those two things in mind, how do you envision actually combining them? So it's such a complex question. I'm having trouble putting it even into words.
Kelly. So it is a really good question, and kind of central not only to this project but to the whole field. There's so much complexity there, which is why I moved into this field, is because there's just so much to discover. And this is full employment for the entire planet's population for the next couple hundred years. And so we really have a lot to ask. And I hate to say this, but we have to get reductionist. So one of the things that I think is misunderstood about all the omics is the omics are collecting all the data at once, and that's certainly correct. But the intention was never, I was here at the founding of this field, the intention was never to try to study everything all at once. So prior to the omics, you were in a situation where, and I go back that far, in the 70s or 80s, we studied something because we knew it was there. We said, okay, I'm going to study, my colleague who was studied spore proteins in yeast, and he studied it because he knew it existed. And he didn't have any genetics on it at that time, even though it was yeast. And so he didn't know if it actually did anything, but he knew it was there, and he's a biochemist, so he just studied it for years. And it was a really major protein that's expressed during sporulation in yeast, and so he was really excited about it. And then after a few years, we started doing knockouts in yeast, and we cloned this gene and did a knockout on it, and it had no effect on sporulation at all. And, okay, great. This really made this guy happy to spend his whole career into something that had, every gene has a phenotype. You just but not under normal conditions. I'm sure this gene was vital in some environment where sporulation was going on, but he spent his whole career on it. So, and that didn't describe just his project, that described pretty much all of science. We worked on things because we knew they were there. Geneticists had a little bit better angle. They knew what they contributed to because they could get a phenotype out of mutation. But now we're in a situation where we can study everything all at once. And the purpose of doing that is not to study everything all at once, but in fact to find the most interesting correlations from this sort of descriptive omic work, and then say, okay, this guy really lines up with this guy, and this looks really important. So if I find, for instance, a regulatory gene that's involved in determining whether AMF is present, I'm going to personally find that much more interesting if I find a structural gene that determines whether AMF is present. And so I'll study that one. But so we can get really specific by mapping QTL, and the GWAS, as you know, despite its outlandish level of false positives, does, when it gives you a real positive, gives you a really super well-defined one, so you're often to the gene, much better than a normal QTL mapping would do. And so with the GWAS data, we're able to say, okay, this is the gene we think is important, and this looks like an interesting gene. And that's really sharply defined. Now, the microbes it interacts with, that's going to be a muggier thing, because the trouble is we think a lot of what goes on at the species level. And if you want to look at microbiomes at the species level, you can't do that very easily by amplicon sequencing, because
exactly.
They give, if you use, it just depends on the kind of sequence you use. It's really hard to get down to the species. And we have cases where we don't think we see a QTL for something because we think we have two members of the same genus. One of them's doing this and the other one's doing the exact opposite. They cancel each other out. In fact, we have examples where we've shown this. If we looked at the two individual species, we'd find two QTL, two different ones. One of them makes this guy do this thing negative and one of them makes this guy do this thing positive. But if you, in fact, put those two in the same data set, there's no QTL, there's nothing at all. So I think microbes are going to be the greater challenge until we can do really detailed analysis, because a species is kind of meaningless in bacteria because of all the horizontal transfer. We really want to get gene to gene correlations. So I want to be able to say it's this plant gene works with this microbial gene, and I don't really care what species that microbial gene is in, although it might be really important, but I don't really personally care that.
Right. Well, that brings up the issue even of having some kind of a genetic map, so to speak, of all the microbial genes, for example, that you could actually get through metagenomic analysis or
when they first sequenced E. coli, the first 3 E. coli that were sequenced, they had 20% shared gene content. The people doing that sequencing figured the value would be about 98%. So they were slightly off in their guesstimate. And so, there's, and if you just look at the number, plants don't have many genes. Even if you look at the entire plant kingdom, there's probably only a couple 100,000 genes. Bacteria have hundreds of millions of genes. If you look at all of them, any given one may only have 6,000 or so, or 4,000 or 2,000 or whatever the number is. But if you look at all the bacterial genes out there, it's dwarfing the number of animal genes or the number of plant genes and all that combined. And so we need really a way to sequence, do shotgun sequencing that's really comprehensive and deep, that we can cover all the genes in a microbiome.
I think that's right. And in many ways, when we look at the history of genomic and sequencing technology generally, we are about 30 years. We're where we were in plants. In microbes, we're where we were in plants 30 years ago.
Yeah.
In reality, because we really don't have that capacity yet to get at all those genes. And not only all the genes, but even when you look at the microbes, we have such limited knowledge of the microbial genes themselves that we're really still very much in the dark age.
The physiology of bacteria, you still want to have a purified bacterium. You can do it other ways. People are doing them in pools, but the information gets really vague. You can't even trace which organism contributed the plasmids that you find, right? When you've got a pool, you can leave.
We can't culture most of them. We know we can
culture the majority, so that's a huge holdup. And combinatorial ways to do physiology in communities, I think that's going to be part of it, but it's never going to be as nice as if you can do them one at a time. And that, that can't be done massively parallelly, which is a way to do this in an automated way until you can grow them.
Right, exactly. Well, sequencing technology is changing, so I'm hoping that as that improves, we'll be able to kind of tackle these. And certainly it has changed, and I anticipate it will continue to change. So another question that came in is, do you have a sense of how many AMF cells a sorghum plant can support?
That's a very good question. We could look at our data and find that out, but we haven't. So I don't know. Cells on the order of millions, for sure, but it may be orders of magnitude too low. Different races in the same plant, once again, and it's very, very high. So it's not like you get AMF infecting here and it excludes all the other AMF, right?
Right.
It doesn't. Other AMF come in which is weird because you'd want that to happen. Both of, certainly the AMF would, from its own evolutionary competition with its peers, it would like to shut down its host once it's got an AMF association, stick with me and don't go with anybody else. But you find them on separate roots. Be interested to know, and I don't know the answer, but we can do this, and we actually are doing it, the degree to which you find different AMF in smaller and smaller areas of the root. How nearby can different AMF be? So, but yeah, that's not a question we know, but it's certainly a lot. It's a whole lot. The very large percentage of the root cells have AMF. Now, that's based on microscopy, and the microscopy is largely limited to finer root tissues, just for technical reasons. It's hard just to actually look at, do the staining and the analysis in a thicker, older root. So we're always looking at younger roots when we do this. But at least, as I mentioned, at least at a community structure sort of sense, not in a quantitative sense, but in a qualitative sense, the different root types seem to have very similar AMF communities. So it's not as if by looking at the fine roots, we're missing a kind of an unusual kind of community that's somewhere else, because it looks like the communities are pretty similar. I should mention, in those partition experiments we did, it shouldn't be obscure to anyone that we're not looking at a tissue. I don't know how many tissues there are in plants. You know, to call a root a tissue is nonsense. There's so many different roots that are doing different things. You know, if I were guessing how many tissues there are in roots, it probably is pretty similar to the number of cells there are in roots. Every cell is a little different from every other cell. And so we are, you know, it's always the case, unless you're doing single cell biology, we're fusing lots of things together that might have very different results, that in our averaging of them, we lose that difference completely.
Well, you've mentioned a term that a lot of people are moving into now, and that single cell biology. Are you thinking about how you could incorporate that to add that little piece of complexity?
We're doing some single cell biology on AMF right now, and that's yeah, both in the plant and in spores. In the plant, it'll actually be easier. The spores are spores. They've got all that stuff around them, and
yeah,
they're big, which is nice. AMF have great big huge spores, but they're tough and rough, and they have all sorts of microbes on the outside of them that's really hard, that are really stuck to them, hard to get rid of them. And so without doing damage, you know, a lot of them are fungi, for instance. So if you say, well, I'll just use some chitinase and get rid of them on the outside there, well, it's fine, except the chitinase eats up the spore too. So,
yeah.
Anyway, we're trying to do some single cell stuff, but it's going to be a tremendous challenge. And I don't, thank God, nothing that we do in the lab lives or dies on whether we not get single cell stuff to work. Thank God.
Right. So one of the things you also mentioned in your talk was the idea that instead of dealing with inoculants, or putting AMF on the plants or in the soil, that you would actually breed for it. You'd put a specific plant genetics in there, but you've also shown that your plant genetics operate differently in different environments. So,
and the starting inoculum of AMF is different in different environments too.
Yeah.
So talk about that. But when we take the soil from these two different locations, and everybody's done this, so that's why I didn't talk about it. I think any AMF person knows this. You get a different AMF community to start with, right? And so that's going to be what you factor in. Is you're going to say, okay, this genotype will attract, is one we want to use in this location, not only because it attracts this microbe, but it also, in this location, that microbe's present to be attractive, right? Whereas this one might work, for other G/E characteristics at another location, but if its job is to attract an AMF that isn't there, then that's not going to do it. So for a farmer to do this properly would require him to know a lot about his soil.
Exactly. Yeah, that's been one of the things that we've been promoting, because if we ever want to get to that vision that I mentioned in the beginning, then the grower really has to know a tremendous amount of that biome. They really have to understand that biome.
And from seed companies that they would love to develop a seed that works everywhere, but they know that doesn't ever happen. And so, Pioneer may sell 20 different seed types in the Midwestern United States. if you add another factor and said, well, no, 20 ain't going to do it, you're going to have 200. Well, then their economics begins to go apart because, you know, there does have to be a certain volume to your sale. If you're selling a maize line you've developed exclusively for one farmer, then that's called going out of business.
Yeah.
Right. So they need to have some general rules show up, and we're a long way from finding those general rules. But they don't need to be, you know, you don't need to find a genetic association that works for all of Iowa. That you do not need.
Exactly. I doubt that you could, in fact. You know,
it's impossible. But if you need to find one that you need 17,000 different ones for Iowa, then you're in trouble. So it's really some sort of place in between. But the thing is, no one's been following these traits anyway, right? They've been doing breeding and just looking at yield at the end, which is a combination of various traits, of which many may have something to do with the root, but they're not ever looking at the root. So it's just a coincidence that they select for it. So when we looked at the Ceteria and saw that all the domesticated lines have different communities than wild lines, it meant that, yeah, exactly, the farmers had been selecting for microbiome characteristics.
Yeah, exactly. Yeah, you know, without even knowing. Yeah. Yeah. So the other thing that you are looking at is you've looked at phosphorus, and of course that AMF is known to have a huge impact on that. But another area that, you know, people are exploring, of course, is carbon sequestration and the relationship between AMF and carbon sequestration. Are you looking at that at all, or do you think you might at some point in the future try to bring that component in?
Yeah, so I said I'm 1 for 1 in microbiome proposals. I'm 0 for 1 in sequestration proposals.
Okay.
which
so was right.
That's 15 years ago. So, and the basic was the roots and the microbiome should be able to capture a lot more DNA. You know, the trouble at that time, and it still tends to be true, is most of the carbon sequestration money is PR and con jobs, and you know, this, oh, we're going to pump CO2 into the ground. I mean, it's just nonsense. It's sort of like algae was for carbon capture for biofuels. The reality is plants do this, and they do it super well. But carbon that plants pump into the ground can be very rapidly metabolized by the microbes there, and so. But some isn't, and so, you know, probably most of the carbon, I would guess, in the soil that's captured there, I don't know this because I didn't really follow this field up, but at the time, it's actually the root degradation products with things like lignin that have really long.
Yeah, I think that's true. That's changed a little bit with some of the, at least what we're beginning to understand is more, the microbes actually play a greater part than plants.
Yeah, but they play a part in both directions, and so you basically, they do mineralize sometimes and make stuff more stable in the soil. More often they do the opposite. So studying that is going to be really important, really complicated. And there's a lot of room for those studies, but I think the potential is enormous. We are not doing anything on it now. We have collaborators who've contacted us about doing it, and we just haven't decided whether we can.
Yeah. It's early. We've still got a lot of research left to do. So.
Yeah, well, we're just, you know, this project, I gave you sort of the scale we're talking about here is, it's a lot of people working a lot of time. So, you know, I'm not sure with my limited brain cells, I can really handle making this more complicated than it is now. So I'm always happy to find collaborators who will do almost all the work. That's my favorite collaborator, but unfortunately, it rarely turns out to be that way. Sometimes.
Yeah, that happens. Well, Jeff, thank you very much for a great presentation and an interesting project and a good discussion. And I look forward to watching this project as you advance and as we make these kinds of changes that are obviously will be positive for agriculture, but not only agriculture, but for humanity at large, which is what you've been working on for your entire career. So.
The goal, yeah. So thanks.
Just a reminder, the webinar has been recorded and will be available on our YouTube channel. So thank you again, and we look forward to our next webinars. Bye-bye.
Bye-bye.